I hope it is recording. Okay, just in case, let me put this here as a backup because I know we'll not have time to go back and talk again. Okay, we're good. All right. Okay, so in this case here, if you look at the karyotype with in situ hybridization, you're going to see that there is a piece in the chromosome number 7. Then you see this is a control here, and you see the probe here. You're supposed to have 2 of these hybridization sessions, and then here you have only 1. So there is a deletion. You see, in the normal case, you have 2 hybridizations in one chromosome and 2 hybridizations in the other chromosome. And then here, 1 is missing. Now, by looking at this, and you see, okay, this is a person with Williams syndrome. This is one chromosome, and this is the other. So if you have here only one that is missing a piece, only one of the alleles is missing a piece, and you have all the phenotypes of Williams syndrome. So what type of dominance is that? Only one of the alleles here is defective. If you have both of them, let's say A, they are there, let's just call it A just to make it simple. So you get the phenotype, okay, or it could be this or it could be that. Now if you have this and you get the phenotype, this is just like plain dominance. You get the phenotype if you have one of the alleles. So it's not recessive. It's just a dominant effect. Now, what happened to these people is that they have these specific facial characteristics. You know, the mouth, the upper lip is a bit turned, they have a big mouth, a big smile, the eyes have a certain characteristic, and the nose. And they basically all have the, you know, they have these facial characteristics, they're all the same. Just like you can distinguish a person with Down syndrome, you know, that with characteristics there. These are the Williams syndrome, Williams people. Now, what happened is that these people are very friendly. They do not fear strangers when they're kids. They have no inhibition about, you know, they're party animals. They love to go out and play, and they're very pleasant people to be around. And of course, you know, this type of chromosomal problems, you always have some type of mental effect or delay in the developmental stage. So they are a bit behind. They'll come and they'll talk to you, and they'll make friends very quickly. But after a while, they just run out of things to talk about. And this is one of the characteristics of the Williams people. And another thing that is extremely interesting about this syndrome is that these people can learn language like nothing on earth. There is one lady, I have to go to the site here, that and music. They just have the ear for music. They love music, they learn music very quickly, they compose, they sing, whatever, you name it. And there is this one lady here, there's some sites. I never watch TV, so I don't know. One second, let me see which one. No, she does not have Williams syndrome. No, I think she has some type of problems, yes, but I don't think she has Williams syndrome. I'll show you here one person. This lady, she sings, I think, in 52 different languages, fluently. It's amazing. So interested, I put some connections. Oh, this is the lady right here.
I arrived on Tuesday evening when there was a concert. Just as soon as it began, everyone there responded to the music.
So this is a camp for Williams syndrome patients. These are people who are really party goers. Now let me fast forward a bit. You know, you can go back and watch the whole thing. Well, oh, this lady here. She's the one that sings in like 52 different languages. And since an early age, they just have this ear for music and rhythm, and they just compose, and it's really amazing. And now, why does that happen? Who knows? Just a little piece of the chromosome of one of the alleles that is missing? Yes?
Well, if that's the case, why don't they just say, like, I don't know, maybe the brain of these person people and compare them to prodigies and see what the difference is?
Well, that would be a cool thing to do. I don't know why.
Maybe they, what if find the comments how it was doing them so great and here's things.
Yes, it's true. It's true. But I don't agree 100% with that. No. Okay, I'm the oddball. I never liked language. I totally hate English when I had to learn it. I mean, I grew up in Brazil. First, I hated Portuguese because my family is Italian. We always spoke Italian. And then they put me in elementary school and everybody spoke Portuguese. And I was totally out, a fish out of the water. And then, after like 14 years old or so, and my mother decided to move to New York. And there we go again. I mean...
Well, it doesn't mean that you like it, but it affects the similar areas of
Yes, but usually people who have this type of a syndrome, they have facility for learning language, and they learn very quickly. Now, when you have a lot of musical talent or tendency or facility to learn music, you don't necessarily have a lot of facility to learn language. Okay, yeah, you might activate sort of similar areas, but some other people have those areas totally isolated. Yes.
Will the correlation be with a little bit their mental retardation? Like children pick up language very quickly, and as you get older, it becomes harder. So with that, maybe it has to do with them being a little bit slower.
No, I think that has to do more with brain plasticity. And it is still possible to learn after whatever age, but then the neurons do not make the connections as fast as children. Children are just sponges, They have nothing in their brain basically, so they have to fill it up with something, whatever comes across. Yes. So, but it's very interesting. Going to some of these sites and check out about Williams syndrome is really a cool thing to do. Now, one thing also before going on, I was listening to one of the lectures of my favorite professor in college in New York. Mikia Kaku. He is Japanese. Have you ever heard of him?
No.
Never heard of Mikia Kaku? Yes, the physicist. Somebody heard of him. Physicist. I think so. As I said, I never watched TV, so I wouldn't know. He has kind of white hair, kind of shoulder length. He's a fun guy to be around. Yes, he's a physicist. He's a physicist. Actually, it was funny that when I was taking his class, I was first a physics major, and then I got to his class, and he came to me and said, "No, you have a biological brain. You don't have a physicist's brain." And I was thinking, "Oh, gee, is that an insult? I mean, I'm doing well in the class." And he said, "Go into biological field." I said, "Okay." I decided to take his idea since he was ranked as one of the 100 smartest people, So I said, he must be right on something, But the one lecture that really stuck to my head was one time when he was saying that if you think about the progression on what we know now about genetics, and you compare it to the internet. For example, when the internet came up, it was like around 1982 or 83, something like that. And how was it used? It was used as a way of defense for the Cold War, right, against the Russia, Soviet Union, something like that. It was a very secretive weapon that the United States had in order to communicate, and I don't know what they were using it for, but it wasn't a war-based principle. Okay, actually, at the City College of New York, there is this guy who actually came up with the fiber optics. And he thought there was no use to it, so he just sold to the government for like pennies. And yes, fiber optics. And he said, what's the use for it anyway? So, exactly. Sometimes you come across these things that is like, but that's when the internet was starting. The idea was it was a weapon of war. Now, after Soviet Union, came down, everything changed, and then the internet is out there. And if you look at the internet now, it's not quite used as a weapon of war. Okay, and it was very funny that he was mentioning a few years ago that 25% of the usage of the internet is for porn sites. Okay, so he said because usually you have at that time a lot of young men who don't really have a lot of fast access to these very interesting things, so they go on the internet and they're like get all fascinated by all this anatomy. So he said, and this is because a lot of the population using the internet is made up of this composition. He goes, now you just wait until the elderly population really gets familiar with the internet. And he said, and then you're going to have 50% porn activity, like of total activity. And you're thinking more intellectual, more this and that, but he turns the thing around. It's very interesting. And then he came with the analogy with what you know about genetics today, because we are really, really at the tip of the iceberg. And as you have seen with the Gattaca movie, we are not very far from that reality right now. And one concern that is always in everybody's mind is genetic discrimination. If you go apply for a job, are they going to just test your blood and check if you're going to have a heart attack in a couple of years? And what are the chances of you getting the job or not? Actually, real life happened last year, 2 years ago at the College of Pharmacy. This guy was hired as the assistant dean for pharmaceutical sciences. Great guy, great scientist, great brain. 6 months later, he had a heart attack and he died. And everybody's just in shock. He was just starting. Everybody was just, how can this happen? And we were getting together, and when they told us that he passed away that morning, there was silence in the room, complete silence. And sometimes we say things and we may regret later, and I do have a big mouth. So at that time, I said, well, but he cannot die, he just started. I mean, what is going to determine if you can or if you cannot die, really? But is genetic discrimination going to be a reality?
Don't they kind of do that now with insurance, health insurance?
They're not supposed to. They are not supposed to. But what is happening in the background, behind the scenes, you never really know. But
if you already have a
pre-existing condition, that's what they use. But it's not genetic discrimination. The genetic discrimination is going to be, even if you don't have any symptoms of whatever you're supposed to get,
right.
I'm not going to insure you because you're going to get it. That's the genetic discrimination part of it. Well, chances are. Chances are. Right. But that's, then is when, that's when you come to this question of what is really that makes a person. I mean, I really wish the whole class had gone, but I didn't know about this lecture until last week, because this guy, and he's very brilliant, this neuroscientist. He's very, very brilliant, but he is really a shell. He does not believe on anything else other than neurons making connections and neurotransmitters. And I don't know about you, but I do believe there are more things out there. And I also heard, at that time that he just had a baby, and I thought, okay, let the plasticity of the neurons change once this child grows. He'll change, no problem, if not in this life, in the next one. So, but the whole idea of what makes a person and what is really shaping you as a human being and what shapes your thoughts, I do not believe it is only chemicals and DNA and neurotransmitters and neuron connections. So this is where we really have to come to the, understanding of what is moral and what is ethical and where to draw the line. No, but just to keep in mind. So coming back here to our mutation, one thing that is important to know, and I had also mentioned this doing the labs. I don't remember if I mentioned both labs or not. Coming to the DNA technology, how you can study the effects, so how can you study how a particular gene is working? One thing that is very common, very popular to do is site-directed mutagenesis. Now, how do you do that? Well, first you have to find out where in your protein that is the active site and what part of the protein is the most interesting part that you can study on the job. So first, let's say we have a protein here, which, by the way, this could be a nice test question.
Oh, you should get the flu shot, like whenever I go to the doctor or to the doctor, you should get him a flu shot and so you can get sick after that, especially if you're already sick.
Yes,
no,
no, if you are sick, you should never get a flu shot.
Yeah. So he said that wasn't good.
Oh, really? Okay, let me get it. Okay. No, I tell you, no. They say, oh, yeah, this doesn't happen, that doesn't happen. But after you get sick and you get, like 40 degrees fever and no Celsius, of course, and in bed and you feel miserable and say, I shouldn't have taken. I'm not advocating for not taking the flu shot, but if you're not in the risk factor, if you're not, elderly or health problems, asthma problems, etc., and you can avoid, just stay close to anybody who has the flu. Just I prefer to go the natural ways. The flu is not really that great or shocking. Yes. I am not sure about a neurotoxic vaccine. I don't know, because that is a product. That is a chemical. Unless you can make T cells that will degrade that chemical to an inactive form, that might be it, but I don't know really. Because once you have some toxin in the body, it's very difficult to get rid of it. Yeah. I don't know where they're going to get their subjects. Oh, please. Oh, we have to draw the line somewhere, right? Okay, so now we know how to do site-directed mutagenesis. Mutate your primer and you get your mutated protein. Then you study whatever the protein does. Another way to study proteins with DNA technology is by making transgenic animals. What that is that if you have a mouse that does not express a certain human gene, which the mouse is not supposed to express a certain human gene, but you want to know what the gene does in a living system, you can just get that gene and insert into a fertilized egg, insert into the nucleus, and then implant it into a pseudo-pregnant female and let the mice pop out and figure out which mouse got your gene. Once you identify which is the mouse that is expressing your gene that you put in there, that is called a transgenic mouse. It is transgene because it's not supposed to be there in the first place, something from the outside. Okay, so you transferred something into that mouse. Oh, no. Yeah, it's very sad. It's very upsetting. But especially when they're babies, you get all these mice and you clip pieces of the tails off to do DNA, they digest the tail and analyze the DNA, run the DNA on the gel, do PCR first, run in the gel, and the ones that didn't make it, they just, put a little too much anesthetic, they just sleep forever. So, at least with anesthetic is okay, halothane is fine. I mean, you can kill horses with a 1 milligram of halothane, basically. But I do have a problem when they put these poor little mice in dry ice, because CO2 is also going to put them sleeping forever. So, and it's cold too. And poor thing, at least let them die comfortably. But yeah, the ones that don't make it, they're just disposed of. I don't like animal research, by the way. I don't have the courage to do that. Sometimes I have to, but I feel sorry for them. Okay. Now, and also, yeah, if you're going to do animal research in your life, try not to feel sorry for those animals, because if you do feel sorry for them, they take a lot longer to die for some reason. I just don't know. Maybe it's that effect that the water will boil, at a slower pace if you look at it. I don't know what it is, but I know that, my friend from UM, Agatha, whenever she had to sacrifice mice, she used to tell me to leave the room. So, because she said her mice will never die if I'm in the room. So I said, no problem, because she said that she just use more anesthetic and, decapitate and truncate whatever the mice, and I said, just go. Oh, happily, go. But, one of those things that does not have to do with neurotransmitters, and maybe has to do with brain waves that synchronize with the mouse's waves. Anyway, coming back to it. Knockout mice. So, knockout mice, what do you do here? You have a gene that is important for all species. Some genes that you heard about, important for all species, are genes that make ribosomal RNA, tRNA, lots of messenger RNA, and nuclear proteins, nuclear lamina, and transmembrane proteins. Any of these genes that are very important for life, and you want to study the function of it, what you can try to do is to make a knockout mouse. So you get the gene, and you put site-directed mutagenesis. There are many ways of doing a knockout mouse, but the easiest way is do site-directed mutagenesis right in the beginning of that gene, and you put stop codons in there. So that can be done, but that's more for transgenic mice. That, when you want to implant the gene in there. Now, when you want to knock it out, you just put stop codons, or splice the gene out, cut off and ligate. And then you see when your pups, if you do stop codons, you're going to see still the DNA is going to be there, but the protein is not. And when you just cut the gene out, you know that you do PCR, the gene is not going to be there, and still the protein is not. So, knockouts anyway, knock out the function of the protein, knock out the protein. So then what you do, you select for the mice that are not expressing the protein, and you see how different they are from all the other mice. Now, you have to keep in mind here that making knockout mice is not possible for all the genes, because if the genes is absolutely essential for life, you're never going to get a knockout mouse. In fact, the best you're going to get is a heterozygous mouse that has one allele knocked out, but the other one is functional. Still, that mouse is going to express less protein than normal, but it's still going to be alive, so the function of one of the alleles is sufficient to support life. Okay? And if you have in mind that picture of that mouse that had BubR3, which is a checkpoint protein for cell division, BubR3 that was knocked down, so it had a heterozygous mouse. The mouse got older a lot faster than any other of the mice. So they know that those specific checkpoint proteins have to do with cell division and aging. That's how they were finding out the function of some particular proteins. So, transgenic, knockout, not talking about cancer, yes? 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 Why you have to breed the mice again? Because the mice actually have a very small lifespan, and when you are working and trying to figure out the function of a particular gene, you take a long time to figure that one out. So you have to have a lot of mice. Yeah. So you expand based on the one that was your phenotype. Your genotype, your phenotype. So you just keep a lot of them in stock. Okay. Oh yeah, another funny story I tell you. My friend, right, UM, she was doing work with P53, which you're going to come across for cancer. Okay? P53 knockout mice. And there was this other guy that totally hated her guts in the lab. And she had to get the mice for her experiments from this guy that had access to the animal room. She could not just go there and pick up the mice whenever she wanted to. He was breeding the mice, okay? So on a Friday afternoon, 4 o'clock in the afternoon, he comes with 32 mice for her to, sacrifice and get the hearts out. You know how difficult it is to get the heart of a little mouse? It takes a lot of time. And then you preserve it, and you have to do all this, this whatever process you did. And then he comes with the mice. Worst of all, he brings the mice in a paper bag, and he puts it in the sink, in the wet sink. Yes, we had a mice escape at the lab.
Really wanted to escape.
And the strangest thing you can imagine, okay, this poor lady is there and she feels something crawling up her pants. So very quickly she just takes off her pants and starts to shake. So I go in there and say, no, Monica, what are you doing? There is a mouse in here. So he left. Eventually, we never found the mouse. He showed up a week later in the secretary's room somewhere. There goes the knockout mouse. But people say that life in the lab is not exciting.